Ja. Ja, maar heb je de testlinks? Die zijn het laatste gestuurd. Zal ik ze even doorsturen? Ja, ik kan je horen. Ik ben even... Ja. Nee, maar ik ben benieuwd als je de live webinar nu weer opnieuw kan beginnen. Ah, oké. Ik dacht dat je zei dat het al begonnen was op de live webinar, omdat dan... Oké.
During this webinar, in which we're going to discuss a very new, exciting paper. But first, let me remind you that we have our vision to connect. Oh, sorry. To connect global expertise and leadership in order to decrease the global burden of RSV infection, with the mission to support performance of high-quality research, to improve knowledge of RSV epidemiology, and to develop safe and effective therapeutic and preventive intervention. With the goals to advocate and create awareness for RSV disease, to act as a focal point for effective partnerships with stakeholders, and to combine knowledge and capacity required to enhance development of novel RSV therapeutics. Why RSV research and education? And as you all know, RSV causes so many diseases, so much morbidity and so much mortality, with 120,000 deaths, most of them in the first 5 months of life, and most of them, virtually all of them, in low and middle income countries. You're part of the ReSViNET webinar series, in which we have the objective to promote knowledge sharing of latest development. Really exciting, groundbreaking study that really put the science to a next level, and to provide education to professionals in the field. We do this bi-monthly, and we may even increase our frequency of presentations because of the interest that you all had in the past with the webinar. Today, Professor Lopez will talk about the detection of defective viral genomes as a potential tool to predict RSV clinical outcome. And Dr. Lopez will be introduced in a second by Ulla Buchholz, my co-chair, who is has been an RSV vaccine expert, in particular the live attenuated vaccine developments over the past years have all gone through her hands. In a second, I will hand you the microphone, Ulla. So what have we done so far with our webinars? I will be very short. We have had several presentations. First, you got the toolbox from Fabian Sisterhan to make your own vaccine, an exciting science paper. Then Shabir Mahdi from South Africa taught us about the Novavax trial results. Then Peter Openshaw used his human experimental model to instruct us how to understand neutrophil inflammation and its role in disease severity. And last time, Dan Weinberger from Yale explained to us how the COVID pandemic affects RSV dynamics. We had very good patients, and we're very proud of that, and that led to ambition to further our webinars to maybe more frequent and even better webinars. Without further ado, I'm now going to hand the microphone to Ulla, who will introduce our first speaker. Ulla, the floor is yours.
Thank you, and welcome everyone for the ReSViNET seminar series. It is my great pleasure to introduce Carolina Lopez, professor and BJC investigator in the Department of Molecular Microbiology at the Washington University School of Medicine in St. Louis. Carolina obtained her PhD from Mount Sinai School of Medicine. She studied dendritic cell responses to respiratory viruses and respiratory virus immunology in the group of Thomas Moran, and then joined the faculty there as a research professor. In 2010, Carolina founded her own lab at the University of Pennsylvania. In her research at UPenn and now in St. Louis, where she moved in 2020 in the middle of the pandemic, she elegantly combines RSV molecular virology and immunology. Or more specifically, she's one of the very few people currently working on the role of RSV defective interfering particles, which are like virus particles with defective genomes. The research on DIs of our RNA viruses goes back to the 70s when Dan Kolakowski and Laurent Roux studied Sendai virus and Jacques Perrault and others VSV. And at that time, DIs taught us a lot about virus replication and transcription of negative strand RNA viruses. And they actually accelerated, those studies accelerated the development of reverse genetic systems. And DIs also were recognized really early on as strong inducers of interferon antiviral responses. And more recently, thanks to Carolina and others who now study DIs with like cutting-edge modern technologies, we are learning that DIs exist in the natural host. And in case of RSV, Carolina's work shows that they may have a role in pathogenesis and immune response. Carolina's own research on DIs has moved from really basic science, beautiful basic science, to exciting translational work, including her most recent Nature Microbiology publication. And I'm really looking forward, Carolina, to your fascinating results and how you elucidate beautifully the role of DIs as predictors of the clinical outcome of RSV disease. Would you like to take over the screen?
Sure. Okay, thank you so much for having me here, both Louis, Ulla, and every support person that have worked with me through this. So it's exciting to share with everyone our latest piece of work that it's been actually cooking for a long time. So I hope I can convince you that we're learning that these defective genomes are something that we really need to pay attention to, and maybe they're helpful in the clinics too. So let me see somehow. Okay, so just to put everyone on the same page, I'm going to define defective viral genomes as truncated or modified viral genomes that has lost the ability to self-replicate, but retain the ability to be replicated and packaged in the presence of a helper or complementing virus. So this is important because we're not talking about dead-end products that are occurring during replication of RNA viruses the whole time, but we're talking about products that can be packaged into particles and impact the environment, right? Next neighboring cells and the immune response, etc. So this is this is an example of how they look. So this is a Sendai virus. This is a paramyxovirus of mice, and we really use these a lot as our tool to understand the defective genome biology. And you can see here labeled in green are defective particles. And this is a particle that is, enveloped, looks like a normal particle, but it's smaller inside because it's carrying these very truncated genomes. So as Ulla mentioned, the research on this started actually in the 40s in identifying defective genomes in influenza virus. And I'll show you later, this has been described in every single virus that people have looked for them. But how do we generate them? So I'm just going to very, very simplistically summarize this in this slide. So on top in A here, you see that the polymerase, when it's replicating these genomes, can make mutations, right? So we hear a lot about these variants in the current COVID infection. And it also occurs in RSV replication. So you get a lot of variants, and in some cases, these mutations really severely affect or alter or eliminate a particular viral protein. And in this case, the virus now is not competent to replicate on its own and needs to be complemented. So this would be an example of a defective genome. You have also deletions in where actually the polymerase skips full pieces of the genome when it's replicating, jumping forward and continuing replication. So again, now you obviously have the generation of maybe truncated proteins or completely eliminated proteins. And this is very common in some virus families. It also occurs in RSV, also is not as frequent as the next one, which are these very, very severe truncations that we call copybacks or snapbacks, defective genomes, in where actually the polymerase releases from the template when it's replicating the virus and binds back and copies back the genome. So these are very impotent, or there's no transcription from these kind of defective genomes, that they are flanked by the antigenomic promoter. So they contain all the signals to be replicated and packaged by, complementing proteins from a co-infecting virus, but they are not actually producing any protein. So these are also highly interfering because they're competing with the proteins of the virus. So they also reduce the virus replication itself. So as I mentioned, these have been described for every single virus, RNA virus that have been looked for. So you can find all viruses here, but the focus today is on RSV. And for RSV, they were first identified in 1982. Again, in this paper, just as a description that they occurred during replication of the viruses in vitro. There's a couple of other papers here that were published early on, basically describing how these defective genomes impact, for example, the promotion of viral persistence in vitro, which is a property of these defective genomes that I don't have time to discuss now. But really, there was nothing else known when we took over and started to try to understand the role of these defective genomes on RSV. So before I jump into the paper, I'm going to show you a little bit of the evidence we had before that these defective viral genomes in RSV are really, really potent triggers of the antiviral immune response. So as you can see here, this is a very simple experiment. These are A549. These are cell lines in culture that we can infect with RSV that either lack defective genomes, and you see these ones here are the LD viruses, we call them, and it's marked here as low DVG content, so we deplete them for DVGs. Or we can infect them with virus in where we have a lot of these DVGs, or again, DVGs is defective viral genomes, and here you will see this as an HD content, right, high defective genomes. And what I'm showing you in this graph is mRNA, so expression of interferon lambda. Obviously, this is the very, very important early antiviral cytokine produced in response to RSV. And on the other side, I'm showing you the expression of the RSV-G mRNA as a proxy for virus replication. So as you can see here, if you have defective genomes in the infection, these cells are responding with the very robust production of interferon. So the immune system is seeing this virus very effectively, but it's not really responding if you don't have defective genomes. And when you look at viral replication, the RSV-G, you realize that this is not because the virus itself is at higher levels. In fact, in the HD content, in the HD infections, where you have a lot of defective genomes, you actually have less virus. So there is something about these defective genomes that is very inflammatory and induces this antiviral response. If we move this in mice, of course, we all know that mice are not the best system to study RSV, but we move to mice in this kind of, to validate what we've seen. You can see if you infect mice with an HD virus, again, in red here, that would be a virus that contains a lot of these defective genomes. The virus is not pathogenic at all. Versus if you infect with a virus that lacks defective genome, now you have this weight loss that associated with lung pathology. All of this has been published a long time ago, so I'm really summarizing here. So what is interesting is the viruses that do not have defective genomes, the LD, the black line, is more pathogenic because, again, similar to what we see in the cells, there is more virus in the lungs. And in this case, I am showing you again the RSVG mRNA as a proxy of virus replication, but you can see the same if you look at infectious particles. And in the presence of defective genomes, now we have less virus growth. So how is this happening? Similar to what we saw in cells, is because in the hours post-infection, and you can see here, the scale here is 2 to 6 hours post-infection, we already see a robust interferon antiviral response induced in this infection that contains defective genomes, but not in the one that doesn't. So again, validated what we have seen in cell lines. So now go a little bit into the paper. So the first question we asked a while ago, and I think this was an interesting question to us because there was no evidence of these defective genomes existence in natural infections in RSV or any similar virus at the time. So basically the question was, first, can we detect defective genomes in vivo in humans, and do they impact RSV infection outcome? So to do this, and this is the tool that we use in this manuscript, we developed a PCR that can detect these copy back defective genomes in a pan manner, right? So we're not targeting any specific defective genomes, we're actually targeting the whole population. And in some of these readouts, these amplicons, we're actually seeing a number of different DVGs in the same amplicon, right? And again, I don't have time to go in details into the PCR, but I am showing you here how this looks. So you have in green those in the top, those are different patients, and you have in green all those that where we can see signals for defective viral genomes. These are all the copy back type. And versus in black, you see that they are white, right? Or black in this case. So there is no signal. So we could consider those a negative for defective viral genome production versus the green ones that would be a positive. So the way we run this was always run 4 times by 2 different investigators blind to this. And then we actually had these different means to validate and to analyze the data. Either yes, at one of the 4 different PCRs, there was the defective genome. So when we say no, it's because out of 4 PCRs, there was no defective genomes at all that we could detect. The frequency, how many out of the 4 PCRs, and the intensity. So we have different ways to measure this defective genome by this very simple PCR. Okay, so this is the tool we used, and many years ago we asked again the question whether these defective genomes even occur in natural infections in humans. So we did this in collaboration with Rick Hodinka, that at the time was the director of the clinical virology lab at the Children's Hospital in Philadelphia. And we just went and asked him for samples that were positive of RSV. So these are nasal lavages that they take as a confirmed diagnosis when the kids made it into the hospital. And they just they store them in freezers. So they were banked samples taken for the purpose of validating, or identifying the infectious agent in these people. So we asked them to give us samples that were positive for RSV A, and that has equivalent CT values for viral load. So at least we could control that the viral load itself in those samples were similar. And to our surprise, because again, there was no precedent for these, we identified in these samples DVGs in 49% of the samples. This was really, really exciting, again, because it was the first demonstrations that these actually occur in humans infected, in nature. But even more exciting was that when we analyzed in these respiratory lavages for the presence of inflammatory cytokines or antiviral genes, we can see that there is a correlation between the amount of defective genomes we found and the expression of antiviral genes. So again, this is a real crucial, and it was a real important moment, I would say, in the defective viral genome fields again, because this field, although it started in the 40s, very active in the 70s, it had completely virtually disappeared because there was no demonstrations that these kind of defective genomes matter in nature. So again, this reinvigorated the field, and for us, this was fantastic because it validated what we had been doing for many, many, many years in vitro and in mice, and it became really important now to understand if they actually impact how these patients do in infection. So this is the next question, and this is done by Johnson, that actually also contributed to the first part of the paper. Jan is now has her own lab at Rochester University. If anybody's interested, she's looking for postdocs, and you know, it's a great scientist following up with some aspects of the RSV defective viral genomes. And she was joined by Sebastian Feld. Sebastian is actually a postdoc currently in my lab that did most of this work. So in order to see if these defective genomes impacted clinical outcome, we had to establish a number of collaborations around the world. And we collaborated with Imperial College, again, probably the same cohort that Peter Oppen had talked before in these webinars, with the Children's Hospital in Philadelphia. I was in Philadelphia at the time, and with Vanderbilt. And I'm going to be describing you these different collaborations and the cohorts and how we analyze this. So cohort 1 was hospitalized children from the Children's Hospital of Philadelphia. So I want to highlight, and I want you really remember that these are hospitalized kids. So these are patients that are highly sick. Right, so they made it to the hospital and they actually were admitted. So these are 122 patients, 0 to 2 years old. Again, all hospitalized, RSV A only, and we excluded premature infants, patients with congenital heart disease, patients co-infected with other respiratory virus, and death. And this is a project that, again, initiated with Reed Kalinka, but then really worked out in a strong collaboration with Chance and Gupta, neonatologists at CHOP in Philadelphia. So what did we see if we analyze whether these patients express defective viral genomes? I mean, this is done 5 years after the other study. Actually, our sensitivity for detection of defective genomes was a little improved at the time. But basically what we see now, if you look at the pie chart here, is in red is the percentage or the number of people that actually tested positive, of these children that tested positive for DVGs. And you see that this is a very large number. We could see DVGs in about 82% of those patients, and in 22 of them we couldn't. If we look at how this correlates with viral load, and the viral load in this case is actually what was reported by the hospital. So we did not revalidate viral load, so we took their CT values, and that's how this looks. And in this case, what you see is those patients that are DVG positive in this case tend to have a higher viral load. Again, this is among hospitalized kids, and I really need you to remember this. So farther on the other end of the slide, you can see this transcriptome that obviously I'm not showing you anything. The point is we also analyzed for how these kids were responding to the virus. And you can see here that there is this very strong signature of type 1 interferon and antiviral and pro-inflammatory genes. So again, in these children, the presence of defective genomes correlates with this enhanced or immune response to the virus. So here I'm showing you an example of these different antiviral genes on top and inflammatory genes in the bottom. And you can see that in these hospitalized patients, the kids that have defective genomes have really much stronger expression of antiviral genes and a lot more inflammation in their, and this is again respiratory lavages and respiratory secretions. So the question was, how does this correlate with clinical outcome? And for these patients, we used two measurements. We use a length of hospitalization and length of stay in ICU as a correlate to clinical outcome. And you can see that the presence of DVGs in these highly sick people actually correlate very strongly with longer hospitalization. And actually, for those that make it into the ICU, it correlates with longer stay. It did not reach a statistic because actually very few of the DVG negative patients actually make it to ICU. So this is all fine and great, but the question is, how do we say that the effect that we're seeing is due to defective genomes and not to the viral load, right? And I have to make a point here that the only viral correlate that has been associated really relatively strongly with disease severity is viral load, but it's not without controversy, since people, and some of you, I think I saw you in the audience today, have seen very clearly that patients that do not make it to the hospital also have a lot of virus. So it's very controversial whether viral load on its own is the cause of the severe disease. So we wanted to ask this question: is the viral load the sole driver of disease outcome? So to do this, we actually look into a different cohort. And in this case, these are non-hospitalized. The majority of this cohort are non-hospitalized children. And this is a cohort that was, part of the Vanderbilt inspired study. And this is, again, a very nice collaboration with Tina Harted that provided the samples and the clinical information. So these are 100 patients, 0 to 1 years old, and most of them are non-hospitalized. We also kept some of the hospitalized to control things internally. Again, we just focus on RSV A, and we actually use only healthy term and normal birth weight infants. So nothing that could be obviously a confusing factor here. So the first question we ask is viral load different in these hospitalized and non-hospitalized patients? And the answer is no. So we have, although we have less hospitalized patients, it's very clear that there is no difference in the amount of virus that these patients have. So it seems very, apparent here that the viral load on its own cannot explain disease severity. Okay, so what do we see now if we focus in the other part, in the patients that actually do better, the ones that are non-hospitalized? So again, in those patients, we see defective genomes in 73% of them, so very large number. In terms of viral load, again, we see this correlation with higher viral load in these DVG positive patients in the non-hospitalized group. And again, we use their clinical data for this. So we are not certain whether these readouts could actually include defective viral genomes pieces and readouts and be altering the viral load. So that's something we need to address further in the future. In terms of inflammatory response, again, we see what we expected, what we're always seeing. You have defective genomes, you seem to have a better inflammatory response, more antiviral genes, and again, inflammatory cytokines. But now, if we look for the clinical outcome, and in this case, they had prepared a score sheet that the parents would answer and determine how sick these kids were. Now, in this case, even though we have all the other correlations, as I showed you in the hospitalized patients, now we don't see that. So in this case, despite having higher inflammatory response, despite having viral load, if that's a real measurement of infectious virus, we don't see an impact on clinical disease in these hospitalized and non-hospitalized patients. So how do we explain this, right? So we're having two very contradictory pieces of data in these two opposite cohorts, I would say. So the one thing that we noticed in this cohort was that actually the non-hospitalized patients were sampled earlier than the hospitalized patients. And again, this cohort, the parents were asked, they were followed the whole first year of infection, and the parents were asked to report any disease symptom within 7 days post-disease onset to the hospital, so then the kids were sampled. So this suggested to us that maybe these early symptoms in the non-hospitalized patients reflected early innate immune responses, because we know that that's what actually the cause of symptoms. And we propose that maybe this was because in the non-hospitalized patients, defective genomes, which are the triggers for this immune response, were generated earlier. So how did we analyze this? We were very lucky to collaborate with Chris Chu, and again in Imperial, that they have this cohort of adults experimentally infected with RSV. They use this cohort for these studies comparing immune responses between RSV and influenza virus, but they were very generous to send us all these samples to us, and we analyzed for defective genomes. So these are 56 patients, ages 18 to 55-year-old. They were all experimentally inoculated with the same stock of virus, which is described here, by intranasal drops. And these were, as criteria, they were healthy, non-smoking patients. So what the whole study was, and the kind of samples we have, I'm summarizing it here so you can have a better understanding of this. Again, these patients were infected with these experimentally infected with this virus, and they were sampled every day for the first 10 days of infection, at day 14 and at day 28. They collected blood for their immune analysis and actually respiratory lavages. So, and even some nasal scrapes. So that's, and also they actually collected clinical symptoms. They have a symptoms score criteria that they use, and people self-report their symptoms. So this was precious, right? Because now we had a longitudinal cohort. I mean, it's adults. Admittedly, it's not the best population, but we cannot do this in children. And they had all this clinical information, and we actually were able to analyze for the presence of defective genomes and correlate this with the clinical outcomes. So in the bottom here, you see how this distribution of viral load in these patients looked, and you can see the virus peaks at about day 7, which is what is expected for RSV, and then the clinical scores also peak at about that time. So what do we see here? Again, remember, these are adults. They have already immunity, so infections didn't pick up in all of them at very high levels. But what we see is actually, despite all these, in 18 of these patients, we could detect defective viral genomes, which was already a good enough number for us to analyze and compare the presence and absence of defective genomes in clinical outcome. But again, our goal here was to study kinetics. So we really needed to figure out ways to analyze this data, because there was no precedence for this kind of a study in this field. So what I'm showing you here in the heat map there is in the y-axis you see every patient, and in the x-axis you're seeing days post-infection there where we had samples, and every blue box is when each patient tested positive by PCR. So what you can see here is that some patients, they took positive very, very early post-infection, some of them a lot later, and some of them had this persistent positive signal. So we just created different cutoffs and where we tried to compare early producers of defective genomes versus late, and we have those 3 different cutoffs here. We just decided that anything that went after peak virus infection, day 7 and late, we would consider late, but we didn't know how early was early, so we tested different cutoffs, and I'm going to show you some of those data. So basically, we're comparing these different cutoffs with everybody else that would be considered late. So what we see, and this, I'm showing you just 2 of these cutoffs here as an example. So if we see defective viral genomes generated first on day 3 or earlier, that's the first 2 histograms here. Bar plots, really, not histograms. You can see in the red color, those are patients that generated DVGs very, very early, and you can see that we can barely see viral load there, and their clinical scores are very, very low. It meaning that these patients actually are doing very, very well compared to everybody else that generated DVGs later, which is in the blue bars. And in the bottom, the cutoff moves to day 5 or day 6. So everybody day 6 and before, we still see this tendency. So the earlier the DVGs were first seen, actually, the better these patients did. So this is just the statistics, again, and all through the course of infection in the bottom, and you can see in orange here those early patients, and this is actually the cutoff of day 5, but we obviously see the same and even more significant if we look at day 3 and earlier, compared to patients that did not produce DVGs, the gray line here, or that produced late. Then the other analysis that we did was, now early versus late was clearly a difference, but now we saw these patients that in where we see this, what we call prolonged or persistent defective viral genome generation, and we considered prolonged or persistent anybody that was more than 1 time, and at least 1 of the times passed through the 2nd week post-infection. So the sample is very small here, is 4 patients. However, the data is pretty strong. So these prolonged patients that had DVGs all, through their 2nd week, they actually have higher viral load, and they have a higher disease score. They also have higher inflammatory responses. So we predict that these patients are really representing what we see in our hospitalized kids, right, that are very high viral load, very inflammatory responses, but they're doing worse if they have these responses. So this is the last piece of data, and you can see here now the statistics of these prolonged patients, people that produce these defective genomes very late, are definitely doing much worse than people that just have this transient production or detection of defective viral genome. So in summary, what we observe here, and again putting together everything we've seen from cell studies in cell lines, animals, and humans, is that really this very early presence of defective genomes, copy-back in particular, is very effective in triggering immune responses that are going to manage to control the virus and reduce, in the long run, damage, right, presumably decreasing morbidity. However, if you wait late, right, or if the patient does not develop defective genomes that are at detectable levels until later infection or persistent, this induces an inflammatory response that doesn't seem to be effective in controlling virus, but it also leads to enhanced morbidity. So take-home message, I think the most conclusive thing we've seen here is that the kinetics of this defective viral genome accumulation is really important in determining the clinical outcome of infection, and we do not know what factors determine this. We're currently studying that, and also that detection of these defective genomes can potentially be used to predict worse outcome in hospitalized patients. Okay, so that's a summary. This is the current lab, and I mentioned Sebastian and Jan that were really the drivers of this, and the collaborators that without them we couldn't have done any of this. And actually funding sources also. Money is important. So thank you, and I'm happy to take questions.
Thank you very much, Carolina. This was really fascinating study, I think. Also, I really like that you had the chance and looked so deeply at the different populations, the inpatient, hospitalized, and the outpatient, and then that you got the chance to really look in depth in the challenge study with the timed infection. I think there are going to be many questions from the audience. There is the first question from Mauricio Caballero asking, is a relationship between DVG and pattern recognition receptor activation and the immune pattern of response? So he asked also about Th1 versus Th2.
Yeah, that's a great question. So we have done a lot of investigation into how these defective genomes are recognized by the cell, triggering the immune response. So in the context of infection, where you have virus, right, the main sensors are these intracellular REGI-like receptors, REGI and MDA5, and particularly REGI is a very important sensor of these defective genomes. And not for RSV, although we're doing the work now, but for a paramyx or for parainfluenza virus, we identify that actually the defective genomes form these secondary structures that are unique and not present in the viral genome that actually facilitate the activation of these cellular sensors. I forgot the second question. What was the second part of the question?
About Th1, Th2, if you know if they affect the skewing of the response.
Yeah, so these are actually responsible for viral clearance and in general. So it has the defective genomes, because trigger these sensors, trigger this pro-inflammatory, classical pro-inflammatory response that is going to be more like a Th1, right, or type 1 inflammation. We have the way we've done this is we can extract the copy-back defective genomes and isolate the inflammatory motif and use it as an adjuvant for vaccination, for example. And it's a very, very strong type 1 immunity inducer molecule. So in that context, it's type 1 interferon inducer. We know that RSV has these mixed responses that is complicated, and what we don't know with the RSV defective genomes, again, we're working in identifying exactly what is the molecular structure and how they're triggering the immune response, but as far as we know with related virus, is a very potent type 1 interferon, also type 1 inflammation or Th1 kind of immune response.
So great adjuvant.
It is a great adjuvant, actually. It's really, really good. Yeah.
Self-adjuvant to this, what we frequently say about live attenuated vaccines.
Yes.
Yeah. It's a question from David Tabor. Can you determine the ratio of DVG to replication competent particles during your PCR assays? And is this ratio of any interest, and what would it mean?
Yeah, so, sorry, you want to follow up? Oh, no, I heard something. Yeah, so the classical way, of course, to determine if a viral stock has defective particles is to determine the ratio between infectious particles versus not. So it depends on the virus. You have different tools to do this, but that defines a ratio that then you compare in the different stocks, and the more non-infectious particles you have, it means, or it's again a proxy for having more defective genomes. We have obviously other much more sophisticated ways to do it. We can do it by qPCR, looking at a specific defective genomes, etc. It is very hard with RSV to have a very precise number, because actually, as a consequence of this study, we are realizing that in vivo, in humans, for example, the species of copy-back DVGs that are produced are a lot larger and a lot different than what we see in vitro, in where we favor the accumulation of smaller DVGs. So our viral stocks, because they are generated in virus that will replicate in cells, will have a certain number of defective genomes, but if the defective genomes are being generated in patients, that numbers may be different. So I don't have the answer for what the ratio is now, but we are really working on generating tools that allow us to ask that question right now. And we care a lot about this because, again, I didn't show anything of that, but we do a lot of cell biology and a lot of studies of how the virus interact with different components of the cell and change the biology of the cell. And it's very interesting because some cells during infection, and we see this also in vivo in mice infected with Sendai, which is the paramyxo that we use. In vivo, you have this very heterogeneous composition of viral genomes and defective genomes in cells. Some cells seem to be taken over by those defective genomes. And that phenotype associates with function. So the cells that are taking over by defective genomes are the ones that are inducing the antiviral response and are actually the ones that are surviving longer and triggering in vitro these persistent infections, versus the cells that are never taking over are the ones producing particles, both containing defective genomes or not. So we are very intrigued and very interested in determining what are those thresholds, what make a cell become really, really enriched in defective genomes and change their functions versus not. But again, we don't have the numbers yet. We're working on trying to determine that. Yeah.
Yeah, that is probably my own question, like the biggest question that comes to my mind when I read this. All the wonderful, when I look at all the wonderful data, what triggers the generation of DIs? Is there a host antiviral, like, is there maybe just the early antiviral response with a feedback loop or a role in initiating the generation of the DIs? Does it change the activity of the polymerase or the structure of the RSV strand? What do you think, like,
yeah,
the aminase action? You could imagine a lot of things, and you probably know a lot of things already.
Yeah, it's a fantastic question, and we actually have a whole project right now in the lab trying to figure out what are the host factors that determine this. And we do have evidence that I didn't show in ex vivo. If we use a transplant from people, so we use different people lung tissue, but the same virus, right? The same virus as stock, so we don't have virus as a variable. We clearly see that in some tissues, in some people, defective genomes accumulate a lot earlier and others very late, or you can't really barely detect it. So there are some host factors here that we don't understand. So many years ago, we asked whether the antiviral response itself was responsible for triggering defective genomes. And the answer is actually very clearly no. In fact, defective genomes are the ones that trigger the antiviral response. If you don't have them, you don't have antiviral response. And that we've shown with many different viruses. And if you don't eliminate, let's say, interferon or, interferon receptor or anything like that, in fact, you have higher viral load, and the defective genomes eventually, because they will be generated at some point in infection because they're higher replication, they are actually at higher levels. So it's not in response to the antiviral response or interferon as interferon itself, but we are really looking into factors because there's definitely factors that we haven't really been able to determine. One of the interesting things of this study that we just did, we looked for a number of demographics, for example, information that we had available. And the only thing that seems to be consistent among cohorts is actually that females generate more defective genomes. And we need more data and have more power studies to determine whether that means that females are doing better or worse with DVG. I cannot answer that question. But just in terms of their detection, in our ability to detect the defective genomes at any time during infection, it seems that both in children particularly is very strong, but also in adults, females generate more defective genomes. So, these are the only things that we see, but we're really working on this. Yeah.
That relates to a question by Emily Pfeiffer. How can we detect DVGs at the pediatric ward?
Yes, I need to convince people, anybody interested come to do this, I need to convince people to start developing assays that are, usable, right? Because these PCRs are, just not for the hospital. But if we are thinking, in how we can think on ways in where we can have a testing mechanism to be used in the world. I cannot tell you now what the answer would be. Yeah, but someone may be interested in developing it. I think it would be really neat. Yeah.
Here's another question by Ashley Weaver. Very interesting. She says, are higher lower DVGs associated with specific RSV strains? Or let me expand that to like subtypes A or B. Are you looking at both or only A?
So far, we only looked at A. We are interested in looking at B, but again, for me, it's a matter of manpower and having enough people interested in doing these different things. As you can see, we do a lot of everything from molecular biology to this, so there's always a lot of interesting questions. And it's being, the follow-up for this study is actually looking at B. We do have actually some B samples in the freezer, and see if there is any difference. I don't know, and I think that the reason why my lab hasn't been motivated enough to look at this, and maybe you can help me here, is because there is no real clear evidence that there is any super clear difference in pathogenesis in these two strains. So unless we haven't found that clear evidence, people say, well, we're going to see the same, so why bother? But we can make it exciting for people to look into it. Help me, and I would love to have someone motivated to look at this. Yeah,
I will jump in for a second just to give some air to Ulla, who's doing a fantastic job. Carolina, this was great so far. We have a question from Lisa Glasser, and she is asking whether there is a specific phenotype of people who generate more likely DVGs, and they may be screened for a poor outcome. And let me add whether age might be related, because RSV is so specifically related to age that I was wondering, in addition or as part of this question, whether very young children, probably difficult to study experimentally, are more likely to develop DVGs, or later. Yeah. If I understood correctly, maybe later in the course of disease develop DVGs.
Yeah, it's a great question. I mean, we only have those two cohorts of children. One is up to 2 years old, the other up to 1 year old. So we lack something that we can compare really fairly. We do have an adult cohort, definitely less DVGs in adults, obviously. But I don't think we have enough power data to say anything about the early age. I am really interested in studying that as a factor, obviously. And again, it's a matter of having people interested in doing this, because we can do that, and we can even do that even in mouse models to have a very controlled system with the parainfluenza, because it's very similar, and then use the human samples to corroborate anything we see. So I'm very interested in age, but we don't. So again, beyond females, in the cohorts we did not see a significant impact of age. Again, with the limitations of the cohorts we have. Race was also not a significant correlate. And actually, we do have, especially for the adults, right? We do have the immune phenotype. So people that had more or less IgA antibodies in their, respiratory tract, for example, and or overall immune response, and we actually don't see a correlation with that neither. But again, this is just piggybacking on data that others collected. We will need to do a much more focused study to get enough numbers and the right populations to do that. Yeah.
So let me follow up on that. RSV is typically a neutrophil disease. Also, the experimental model from Chris Chu and the group in London, they have a neutrophil model. They published that, and in babies it. So do DVGs interact or induce a neutrophil response, or maybe depending on the timing of infection? Do you have any evidence from your, host response studies that they induce a neutrophil-like response through interference? There is a strong interference signature in those cells.
Yeah, so we haven't, so studying neutrophils ex vivo is very, very hard, but the nature of the inflammatory response that DVGs induce is highly, type 1 inflammation, which includes neutrophil recruitment and activation. And in fact, with the Sendai model, which we can, again, for mouse studies is a better model because that's a mouse pathogen, grows freely. With RSV, you have to really overload the mice with virus to see things. But with the Sendai model, we have actually seen that there is very, very early recruitment of neutrophils too. So it seems that in these respiratory viruses, the initial inflammatory response, it definitely involves a lot of neutrophils. And that's all depending on having these DVGs. Yes.
Thank you. Before I give the floor back to Ulla to chair the session, Tony Piedra.
Hi, Tony. I see he's chatting here.
He's really trying to give you a hard time. He's wondering how the viral inoculum changes the response of the generation of DVGs in humans particularly. And let me add a small question to that. Do you think in experimental infection, there are DVGs in the inoculum?
Yeah, it's a great question. So we went, it was really, really hard for Chris to find the original stock and for us to get our hand on that to analyze for DVGs, because we actually wondered that, right? I mean, one of the things, just even without telling me what we saw, if you look, there are patients that we never detected DVGs, right? So despite even if there is some or not DVGs in the stock, we do see most patients we did not detect DVGs, right? So when we analyzed the stock, actually we found almost no DVGs. If we actually sequenced it, did RNA-seq of the stock, and we found really, really, really minimum to no DVGs. And the few that we could see, we didn't find as a species in the patients. So we don't think that the virus stock itself contributed a lot to these DVGs. Again, not all patients have, and it's the exact same stock that everybody received. But it's obviously a factor. And I think in terms of thinking on how you may get early DVGs, how you may produce early DVGs, one option is obviously that they are coming in the transmission event, right? That they are actually, you are breathing in virus that may be even co-packaged with defective genomes, that some people propose they can be packaged. And that leads to those very early, events. But this is all speculative at this point. We don't know. But the only thing we know is that viral stock had very minimal DVGs, and the one with the tech we never found in the humans, in the people. Yeah.
Gonzalo de Prat-Gay asks what your thoughts are on the minimal ideal optimal size and sequence of DVG fragments to basically induce various responses, and what sort of therapeutic, or in this case, like adjuvant molecule are you developing?
I'm trying to read. Can you repeat the first part of the question? Because I'm trying to read, he said rephrase, which is the minimal ideal optimal size of sequence of DVG. Okay. So for Sendai virus, which is where we have done most of this, we identified a motif that is 40 nucleotides long, that that is formed exactly where the polymerase breaks and rejoins. So in that region of the genome that is different from the standard virus. That motif we can actually put in a very inert RNA that never stimulates anything and now becomes very pro-inflammatory and a good adjuvant, or we can generate these, we call them DDOs, defective genome-derived oligonucleotides, that we can use to put, together with vaccines and have type 1 immunity. And we really have a significant number of papers on that if you're interested. So for RSV, the difference is for RSV produces a lot of different defective genomes. So the first task was to identify which are the immune stimulatory one, or if they're all of them, or which are the ones that are immune stimulatory. So we're working, a postdoc here now, it's been doing this and using a very small copy-back DVG, he's seeing a very similar situation than with the Sendai one, that there is some sort of motif that forms around the place that the polymerase breaks and rejoin that actually is necessary for this enhanced immunostimulatory activity of these RNAs. So again, this is in a study for RSV, but it seems that around that area there are secondary structures that actually form. And in terms of mechanism, these RNAs, in order to trigger the immune response inside the cell, if they make it inside the cell, it's going to be through Regain MDA5, where they use the 5'3-phosphate. But we have shown that actually this additional motif facilitates binding to these sensors, so it's a stronger immune response. When we use these RNAs as an adjuvant, and we're actually working on this, on finishing this paper now. So now you're actually just injecting it together with the vaccine. Now we know that it's actually triggering through TLR3. So we're just getting it independent on these intracellular sensors, and it's a very good trigger. So that's what we know.
There are questions also about the many different versions of DI. Cyril de Rouen asks if you also find large deletion genomes in vivo. And Thomas Voss asks, have you looked at the genetic diversity of the fully competent RSV between high and low DVG samples? Do they drive evolution in vivo?
Yeah, we are playing a lot with that right now. So we are, one of the challenges, right, is develop the bioinformatics tools that are going to help us here, because the copy back defective genomes, which are the ones that we follow the most, and even the deletions, right, they share the sequence with the virus. So, and it's very hard because these are very structured RNAs to do long read sequencing. So what we are doing is we have developed an algorithm that allow us to identify copy back DVGs, at least the break rejoin region, and with that we can predict the rest of the size. And I mean, we have seen an enormous diversity in RSV in these defective genomes, and a very surprising positions and locations where these breaks are occurring that we are following up. So part of the follow-up is because some of these predicted defective genomes are extremely large, like sometimes almost double the size of the genome, because the polymerase goes always to the end almost and comes back. So we're really wondering if that exists, or is because first there was a deletion. So we're actually, as we speak, developing ways in how we test it and how we identify these species, because definitely there are mosaic situations in where there is a deletion, a reverse, a copy back, etc. But without long reads, it's all very hard to be conclusive about that. In terms of evolution, we are very interested in that. So we are sequencing both viruses and DVGs. We are doing actually experimental evolution right now and putting things like host immune response as pressure and see how that changes the virus and the DVG population. And it's becoming really, really interesting how immune pressure changes the populations of defective genomes, and actually, that is leading us to all kind of weird different directions. But we are actually actively looking at that right now, and also the virus. We find some interesting mutations in the virus that we're validating now because they're really interesting. But
Thank you, Carolina. We're getting close to the hour, and it was so vibrant, so full of energy, and I could. Ulla and I could continuously see each other, and we were both so cheerful. Nobody could see how much energy you have brought to the table, at least to us. I hope the viewers have the same feeling of energy, and very stimulating. You're getting a lot of requests for collaboration, I'm sure, at least from Utrecht. So thank you for doing such a fantastic job. A few people asked us whether they could review or see the webinar today or previous ones again. The answer is yes. So just visit our website and you can see them again. So just to be sure that we close in time, I want to thank you, Carolina, for being here today and showing us this fantastic work that took you more than a few months. Everybody knows how much work is put into such a paper. I'm sure you're getting a good offer for your postdoc. Everybody heard that there is a fantastic postdoc in this field. And I would like to close up with thanking you, Ulla, for preparing this meeting and really putting science to the discussion. Everybody at home, stay healthy. Thank you for being here, and see you again very soon during our next webinar. Have a great day.
Bye-bye.
Great day. Bye-bye. Great presentation, Carolina.
Thank you. Thanks for having me. It was fun. Thank you.
Thank you, Luis.
Bye.